In this tutorial, we will demonstrate how to use the KCIT software simulation to run an expression microarray. This type of microarray measures relative gene expression under specific conditions. So some examples are listed here. If you have cancer cells versus normal cells, or cells that are undergoing a drug treatment versus untreated tissues, or diseased tissues versus normal tissues. So in all of these cases, there will be certain genes that are enhanced and others that are decreased. And that can be used for research purposes or for diagnostics or to determine the prognosis of a disease condition. In order to run this type of microarray, RNA is isolated from the cells or tissues under different conditions. And the reason RNA is isolated is that that is a measure of gene expression, since active genes will be transcribed more in order to make more protein. Less active genes will be transcribed less, so there will be less RNA. And then that RNA is copied into DNA, incorporating a fluorescent dye as part of that process. And typically a green fluorescent dye is added to DNA from control cells or tissues, while red fluorescent dye is added when the cells are in an experimental condition. And then probes corresponding to various genes are spotted onto a chip, and both the red and green labeled DNA is added together to the chip with those probes. And the two different samples of labeled DNA will compete for binding to those probes. And the relative amount of red versus green labeled DNA that binds to each spot can be measured. Green fluorescence is measured, then red fluorescence is measured, and then computer programs are used to combine those data and present an image that's color enhanced, and yellow is used to indicate an equal amount of red or green. To use the KCIT software simulation to run this type of expression microarray, open the software as usual and then find the chip button on the silver button bar. So if you click on the chip button and then open, and you want to select open and load an expression chip file, so then you'll have to browse for the file that you need to run the microarray. And in the KCIT folder that was downloaded from the website, there's a folder called Cases, which contains all the files necessary to run all the cases in the resource manual. If we open that folder and browse to the microarray cases, and we're running an expression microarray, note that there's a single file for each case that contains all of the DNA and probe information necessary to run the case, which is different from most of the cases which require multiple DNA, probe, etc. files. Also, these are CSV files, comma delimited text files, rather than the standard text files that are typical in the other cases. And note that all of the files for the expression microarray cases contain the abbreviation for expression EXPR. So I'll open the tutorial file, and when the file is opened, the probes are automatically spotted onto a chip, and this is on the same window where the ELISA and the dot plot are run. The method is automatically selected, it says expression chip. And there are 64 probes spotted onto the chip. Of course, in an actual microarray there would be thousands of probes, but we've scaled it down to 64. And then over in the Open and Process window, the name of the file is visible, and then the sample that that file contains. And so there's a green labeled DNA sample representing a control type of cell or tissue, and then the experimental cells or tissue is labeled red. To run the microarray, we will click at the bottom of the window here. And then here are the results of running the microarray. So all of the steps involved, adding the DNA, allowing it to hybridize, detecting the fluorescence, all of that has been done behind the scenes after the run button is clicked. And what we see right away are the results of the green fluorescence detection. And you can see different intensities of fluorescence. And the grid to the right of the chip shows the genes that are represented by each of the probes and the spots. They're in the same 64 spot array. And then the value for the fluorescence intensity is indicated. So this fluorescence intensity is almost 3000, and that's a reasonably intense or bright spot. The one below it is about a third as bright, and the intensity value is much less. To view the results of the red fluorescence, you can either click on the red thumbnail up here in the navigation window, or you can go to the Open in Process window and click the name of the red sample. So you can go back and forth between the green and the red samples, and you can evaluate the relative amount of fluorescence intensity for each color. So for example, we're looking at this spot associated with the gene cyclin B1, and with the red fluorescence the value is about 3400. With the green fluorescence it's only about 1200. So in the experimental condition, the expression of that gene is enhanced, is increased. So that gene may have something to do with that condition. If it's a disease, or if we're comparing a cancer cell to a normal cell, that gene might be involved. So you can examine each of the genes one at a time, going back and forth between the green and the red fluorescence intensity and comparing the intensity values. Or you can compare them all at once by calculating a ratio for each one, where the green fluorescence value is divided by the red fluorescence value. And the software will do that if you go to the chip button down in the button bar here and go to compare, and then for an expression chip, what we want to do is combine the red and green data. So if we do that, we'll see a spectrum of colors from red to green, where the red spots, such as the one we were looking at earlier, represent a low ratio. So that means that the red value is higher than the green value, and the green spots represent genes where the green value is higher than the red value. So these are genes that have decreased expression under the experimental conditions. Yellow spots represent genes that are relatively unchanged between the control and the experimental condition. So the ratio between those two fluorescence intensities is around 1. And a rough standard for significance of these ratios is that anything less than 0.5 is significant for an increase, such as the cyclin B1, and anything greater than 2 is considered significantly decreased. So for example, this RAB4 gene here has a ratio of 2.75, so it has decreased its expression significantly in the experimental sample. So you can browse the chip and select spots that have colors or ratio values that pique your interest, and try to identify those genes and think about whether they have significance for that particular experimental condition. But it's handy to be able to group these spots by color, so you could look at all the ones that have increased at once and the ones that have decreased together. And we can do that by going to the chip button and back to the compare for the expression chips and select group combined data according to color. And what this will do is group all of the red spots together so you can see which genes as a group have increased their expression in the experimental system. And depending on the case scenario, you might have information about whether those particular genes have any diagnostic or prognostic significance, for example. If you wanted to return to the original order of the probe spots, or if you wanted to return to a view of the single colors, a green or red color, you can go back to the chip button to compare expression chip and go back to return to view of red chip, for example, or combine red and green data. And then you can navigate again back and forth between the green and the red. But I'm going to go back to the grouped data. Okay, so at this point you might want to find out a little bit more information about the genes that are being increased or the genes that are being decreased in that particular condition. And there are a couple of options for that. If you click on either a cell corresponding to the gene or the spot itself and then right click, it will pull up this menu that will allow you to either copy the probe sequence into the clipboard and open up the NCBI site to the BLAST search page, or you could do a search on the OMIM database, Online Mendelian Inheritance in Man, another NCBI database. You can browse based on the gene name and find out what is known about that gene in terms of human health and human inheritance. So let's first look at a BLAST result. We'll take the DUSP4 gene, which has the lowest ratio, indicating it was the most elevated in the experimental condition, and we'll copy that probe sequence, and this opens up the NCBI BLAST page, and then we can click in the search box, right click, and paste in that probe sequence. And we'll leave all the other settings the way they are. We'll screen or search the entire database and search for somewhat similar sequences using the BLAST N routine, and click BLAST. And here are the BLAST results. We're getting relatively low scores because our query sequence is so short. The probe sequence is only 33 nucleotides, but there are some significant matches, and we can scroll down to the table and see that that probe sequence corresponds to the human dual specificity phosphatase 4. So depending on the case scenario, that may or may not seem significant, and you may need to do more research on what the function of that gene might be. Typically, phosphatases are involved in cell signaling, so that could be involved in a variety of cellular processes. So let's go back and select copying the gene name and then opening up the OMIM site. And because the gene name is stored in the clipboard, you could paste it into any other search site. But we'll click in the search window, right click, and paste in the gene name, and then click. And there are several hits with looks like different forms of this gene or related genes, and again it's a dual specificity phosphatase. You can see from this list that it's specifically associated with ocular function with the cone rod dystrophy condition. If you click on any one of these hits, then you will get a lot more information about this particular gene, what its function is, and here it describes how it's involved in cell signaling, etc. So again, this would be a good starting point for more research into the function of that gene and why it might be elevated in that experimental condition.